In the previous video, we had a look at the structure and functions of biomolecules. We discussed proteins, carbohydrates, lipids, and nucleic acids, their structure and how they function in a living system. In today's video, we're going to have some solved objective questions on RNA and protein synthesis, as well as DNA. Don't forget to like and subscribe after watching, so that you can receive every update from this channel. So let's begin. Question 1. So we have to indicate whether each statement of this is true or false. Question 1. The sugar found in RNA is called a deoxyribose sugar or deoxyribose. Is this true or false? Well, the answer is false, because RNA is made of a ribose sugar and not a deoxyribose sugar. This is found in DNA and not RNA. So the answer for question 1 is false. Question 2. The DNA molecule is double-stranded and the RNA is single-stranded. Is this true or false? Well, the answer is true, because DNA is a double-stranded helical structure, whereas RNA is single-stranded. So question 2 is true. Question 3. The process of translation occurs at the ribosome. Is this true or false? Well, the answer is true, because translation occurs on the ribosome, where the amino acids are being transported from the cytoplasm to the protein synthesis site, which is the ribosome. So question 3 is true. Question 4. The job of messenger RNA is to pick up amino acids and transport them to the ribosome. Is this true or false? Well, the answer is false, because messenger RNA carries genetic information from the nucleus to the cytoplasm, but the transfer RNA transfers amino acids to the ribosomes for protein synthesis. So question 4 is false. Question 5. Transcription must occur before translation may occur. Is this true or false? Well, it's true, because we must form messenger RNA before we can transport the amino acids from the cytoplasm to the codons on the messenger RNA to form the protein. So question 5 is true. Question 6. In the figure below, A, B, and C are the 3 types of. So these are the structures. We have A, we have B, we have C. So what are these? Well, A is a ribosomal RNA, B is a transfer RNA, and C is messenger RNA. So these are the 3 types of RNA found in a living system. So question 6, the answer is RNA. Question 7. Identify the label structures on the following diagram of translation. So if this is a translation diagram, we have to identify this. Let's see. We have this as the messenger RNA. We have the triplet of bases as the codons. We have these ones on the transfer RNA as the anticodons. Then we have the amino acids bonded to the acceptor stem of the transfer RNA. So from this diagram, the part A is anticodon, part B is amino acids, then part C is a ribosome. Question 6. The sense strand of a DNA molecule is this. The sense strand of a DNA molecule is this. The messenger sequence or the messenger RNA sequence from this strand molecule is. So if this is the sense strand of a DNA, then the messenger RNA sequence from this DNA molecule is, well, basically in a DNA molecule, the template strand is the same as the sense strand, and that strand is complementary to the messenger RNA strand or the messenger RNA sequence. The other strand, which is complementary to the template strand, is called the informational strand, the coding strand, or the antisense strand. So if this is the sense strand or the template strand of the DNA, the corresponding basis on the messenger RNA is going to be what? So we have C bonding to gene, we have C bonding to gene, the C bonding to gene, but the A will bond to uracil on the RNA. Then the C will bond to gene, the gene will bond to C, the T will bond to A, the C will bond to gene, then the T will bond to A. So the messenger RNA sequence is gene, 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 U, gene, C, A, gene, A. So that is the sequence which will be on the messenger RNA molecule. That strand is the strand of the messenger RNA. So we have to use the amino acid chart on the last page of this to identify the amino acids from the messenger RNA sequence. So as we said earlier, the messenger RNA sequence is going to be gene, gene, gene. So now daughter, just try it. You have gene, gene, gene. The gene stands for guanine, the C is for cytosine, A is adenine, T is thymine, and U is uracil. So the first triplet of codons on the messenger RNA is gene, gene, gene from here. So let's read the coding table or the codon chart as shown here. So which amino acid corresponds to gene, gene, gene? So we have gene here, we have gene here, then we have gene here. So the first amino acid, we have gene here, we have gene here, we have gene here. So the first amino acid which links these three, this and this, is glycine. So glycine codes for a codon on the messenger RNA, gene, gene, gene. So the first amino acid is going to be glycine because we have gene, gene, gene. The next sequence on the messenger RNA is going to be U, gene, C. So you have U, gene, C. The U is on the messenger RNA, which is uracil, then C is gene, then the gene is C. So we have U, gene. So you have glycine here at the first. So you have the first one as glycine. So the U, gene, C, the U, gene, C on the codon chart corresponds to which amino acid? So we have U, we have gene, then C. So the second codon on the messenger RNA is cysteine, which is U, gene, C. The next codon is going to be A, gene, A. So A, gene, A corresponds to which amino acid on the codon chart? A, we have A, we have gene, we have A. So A, gene, A corresponds to adenine. So we start from here. We have A, gene, then come here to A. A, gene, A corresponds to adenine. So we have the three amino acids. We have the glycine as the first one. The first one is glycine, which is gene, gene, gene. Cysteine is U, gene, A. This one is cysteine, which is U, gene, A. UGC. Then the A, gene A, corresponds to arginine. A is glycine, or 11 is glycine, 12 is cysteine, then 13 is arginine. So that's how to read codons on the codon chart. Let's try it again. Let's say you want to read C, C, C. So the first one is C, the next one is C, so we have C, 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 have C, C, then here the C is this, so we come here, C, which is a proline. Let's say you want to read UCC, UCC, we have U, we have C, then C, so UCC is serine. So that's how to read codons from the codon chart. Let's continue. Question 14. Which of the following is attached to the transfer RNA? So which of these is attached to the transfer RNA? So transfer RNA transfers amino acids from the cytoplasm to the ribosome. So the answer for question 14 is C. 15. Which of the following is not part of protein synthesis? So translation is part, transcription is part, but replication is not part. So the answer is A for question 15. 16. The codon is located on the, so the codon is the triplet of bases that codes for an amino acid. And this one is found on the messenger RNA. It is complementary to the anticodons on the transfer RNA. So 16, the answer is A. 17, in the transfer RNA molecule, which nitrogenous base is found in place of thymine? So thymine is found in DNA. In the RNA, it is replaced by uracil. So the answer is D for the 17. Question 18. During the process of transcription, which of the following is produced? So transcription produces messenger RNA. So the answer for 18 is C. 19. The actual site of protein synthesis is there. So the actual site of protein synthesis is on the ribosome. So the answer for 19 is D. Let's come here. If the DNA template strand reads ATA, then which of the following will be at the corresponding sequence on the messenger RNA? So adenine on the messenger RNA corresponds to or bonds to uracil. Thymine will correspond to adenine. Then adenine again corresponds to uracil. So the answer for question 20 is A. We have AU, TA, then AU again. 21. The genetic code is based upon the reading of how many bases at a time? So genetic code is triplet of bases that codes for one amino acid. So it is 3. 22. Amino acids are held together by, so which bond holds amino acid sequence together? The bond is called a peptide bond, or in some cases it is called an imide bond. So 22, the answer is B. 23. How many codons are needed to specify 3 amino acids? So each codon is made of 3 nitrogenous bases. So 3 amino acid is made of 3 codons. 3 codons. So the answer for 23 is 3. which is A, but it will be made of 9 nitrogenous bases or base sequence. 23 is A. 24. One similarity between DNA and the messenger RNA molecule is that they both contain the same sugar node. The genetic code is based on the sequence of bases. So this one is true. Nitrogenous base known as uracil node and double-stranded node. So the answer is B for 24. 24, the answer is B. 25. Some events that take place during protein synthesis or during the synthesis of a specific protein are listed below. So these are the list of events that takes place during protein synthesis. The correct order of these events are or is. So you have to arrange these events in the correct order of protein synthesis. We have a messenger RNA attaches to the ribosome. DNA serves as a template for RNA production. Transfer of RNA bonds to a specific codon. Amino acids are bonded together. Then RNA moves from the nucleus to the cytoplasm. So which of these is the first step in protein synthesis? Pause the video and try your hands on. So the first one is B. For every synthesis of protein, the DNA must serve as a template for RNA molecule. Then the next one is E. The RNA must move from the nucleus to the cytoplasm. The next one is A. The messenger RNA attaches to the ribosome. The next one is C. Transfer RNA bonds to specific codon. The last one is D. Amino acids are bonded together by a peptide bond. So we have the first one is the first one, B. The next one is E. So we have B, E. So let's come to the list. We have B, E, okay. Then A, as we said earlier, the messenger RNA attaches to the ribosome. Then C, transfer RNA bonds to specific codon. Then D, amino acids are held together by a peptide bond. So the answer for 25 is A. This gives the correct order of protein synthesis. (26) What is the complementary messenger RNA sequence for the DNA sequence shown below? So which of these is the correct complementary sequence on the messenger RNA? If this is a template strand on a DNA, so the C will bond with gene, the A will bond with T, the A will bond with U on the messenger RNA, the A bonds with U again, gene bonds with C, gene bonds with C, then T bonds to A. So you have gene U, U, C, C, A. So the answer is C for question 26. 27. Which process occurs in the nucleus? So we are to use the diagram below for question 27 to 29. So you have DNA giving you DNA, DNA to RNA, RNA to polypeptide, polypeptide to an enzyme, and also to the cell part. So which of these occur in the nucleus? So DNA to DNA is DNA replication. DNA to RNA is transcription. RNA to polypeptide is translation. So 1 and 2, which is transcription, and DNA replication occurs in the nucleus. So the answer for 27 is A. 1 and 2 occur in the nucleus. 1 is DNA replication, then 2 is transcription. The process 2 is known as we said earlier, process 2 is known as transcription. #29. What is the product of process 3? So process 3 gives us a polypeptide, but it must give us first, let's see, a strand of DNA, no. 2 complementary strands of DNA, no. A strand of RNA, no. But a chain of amino acids. For the 29, the answer is D. Okay, so use the diagram below for question 30-32. So this is a diagram for translation occurring in the cytoplasm of a cell. We have to use this to answer question 30-32. Let's see. Structure X was made in the, so X is a messenger RNA which was made in the nucleus. So 30, the answer is A. 31. The process represented in the diagram is mostly or is most closely associated with the cell organelle known as. So this process is associated with which organelle in the cell? Translation occurs on the ribosome. So 31, the answer is B. 32. Which amino acid will be transferred to the position of codon CAC? So codon CAC will have an anticodon gene this codon CAC here will have an anticodon gene gene. So which amino acid carries that anticodon? So histidine will bond to position CAC. So the answer for 32 is D. If a portion of a messenger RNA molecule containing this base sequence, the corresponding transfer RNA base sequence is. So if the messenger RNA contains A, U, the corresponding transfer RNA base sequence is. So the A bonds with uracil, A to uracil, then uracil to A. So that's 3, the answer is D. 34. Which defines a codon? So which of this is the definition of a codon? Well, a codon is a protein that binds a protein that is being transcribed by breaking part of hydrogen bonds. Is false. A free floating base that attaches to an open DNA strand. Is false. A genetic code word of 3 bases on messenger RNA that specifies one amino acid. So the answer for 34 is D. The strong bond between no. So the answer is C for 34. So a codon is the genetic code word of 3 bases on the messenger RNA that specifies one amino acid. 35. What is the role of transfer RNA during translation? So what role does this play during translation? I think we said this earlier. It's bound to open the DNA strand to carry the code for protein synthesis? No. It carry ribosome to the site of protein? No. It break apart messenger RNA and send it back to the nucleus? No. It carries amino acid to the messenger RNA for correct placement into the protein chain. So the answer is D, 45. That is the role of transfer RNA. This diagram shows which cellular process. So this one shows a translation. Translation. The answer is C, for 36. 37. Which of the following changes will be expected if CAU UUG sequence of base is mutated to CAC UUG? So let's come here. If this CAU UUG is mutated to CAC, so this one is a point mutation where one base is replaced. So here the third base, which is uracil, is replaced by CAC. So the only changes here is the change in the first codon. So this one has been mutated to this. So what change can we expect? The amino acid sequence will be shorter than expected. The identity of the one amino acid will change. The identity of more than one amino acid will change. The amino acid sequence will remain unchanged. So let's see. So the second codon is UUG, and it's the same here. But CAC and CAU are different. Let's check from the codon chart if CAC and CAU codes for different amino acid. If they code for different amino acid, then the amino acid sequence will change for the B. If they code for the same amino acid, then the amino acid sequence will remain unchanged. So let's have CAC. CAC codes for which amino acid? You have C, you have A, you have C. So CAC codes for histidine. Then let's come to CAU. So CAC is for histidine, then CAU also codes for histidine. Therefore, the answer for 37 is D, because CAU, CAC, and CAU all codes for the same amino acid, histidine. So the amino acid sequence will remain unchanged. Let's try this one too. So this one, just try your hands on it. The answer has been typed, so you can just pause the video and solve it yourself. It is very blurred, but try and read it. You can pause the video, read, and look at the chosen answer, because the chosen answers are the correct answers. That's the first part. So you can just pause the video and read it. The second part, the chosen answers are the correct ones. The three bases on the transfer RNA molecule that are complementary to one of the messenger RNA codons are called anticodons. According to Chegaff's rule, so this rule says that in a DNA molecule, the number of adenine and thymine are the same, and the number of guanine and cytosine are the same, because The two pairs are complementary. Which nucleotide is always paired with adenine? So adenine pairs with thymine. Ribosomes are made up of what? They are made of proteins and RNA. DNA replication results in two strands or in two DNA molecules, each with one new strand and one original strand. So this one is called a semi-conservative DNA replication. Which type or types of RNA are involved in protein synthesis? So the three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Which of them are involved in protein synthesis? So the three are involved in the protein synthesis. So the answer is D, as chosen. So where does transcription occur? It occurs in the nucleus. Where does translation occur? It occurs in the cytoplasm. So these are the chosen answers and attempt questions for you. So try these questions, solve them, and drop your answers at the comment section. Question 1, the answer. Just attempt and drop your answer at the comment section. Thanks for watching. Don't forget to subscribe after watching so that you can receive every update from this channel. This is Enoch's tutorial. Thank you.